And thank you so much for joining us this evening for the last of our virtual lecture programs that are in honor of the Lehman Caves centennial. This is the 5th program. The other 4 have been recorded and they all were really phenomenal. So next week I will be sending out a follow-up email, and it will have some links in there if you want to catch any of those programs. And my name is Aviva, and I am the Executive Director with Great Basin National Park Foundation. We are the nonprofit partner of Great Basin National Park, and one of our main purposes is to connect people to this park and to help it to be protected and preserved for present and future generations. So we do a lot around education and outreach, supporting the Dark Sky Initiative at the park. We support interns and telescopes. We have a research grade observatory, the Great Basin Observatory, which is the only research grade observatory located in a national park. And we support the BioBlitz, which is really amazing at the park, and just lots of other things with visitor experience and education and outreach. And we're really excited to be connecting you more deeply to the park this year through this virtual lecture series. Tonight, we have Jonathan Reynolds, who is going to be presenting on the Bonneville Cutthroat Trout, which is really a very, very interesting story, which you'll see here in a few minutes. And he has been the fishery biologist at Great Basin for 13 years. We did have, unfortunately, a little bit of technical issues with his screen. So his camera wasn't able to work. What we're going to do, since John and I both are at the park at the Great Basin Visitor Center right now, is he'll go ahead and share his program and share his screen, and you won't be able to see his face while he's talking. Unfortunately, I always like to see people's faces when they're talking. But for the Q&A, we'll just join together on my screen so that we can do Q&A. So basically how we'll go tonight is he'll present for about 40 minutes. During that time, if you have any questions, please hover over the bottom of your screen. There's a place that says Q&A. Just go ahead anytime during the program and put your question in the Q&A. And then at the end of the program, we'll get to all those questions and hopefully be able to answer them all. So the last thing I just do want to say is that the culmination of this very special centennial year is happening this Saturday. So if you happen to live somewhere where you can come join us at the park, there's going to be a lot of fun celebrations happening from early in the morning, 7 a.m., all the way through about the afternoon, late afternoon. So you can learn the schedule on either the park website or on our website, and please come join us. So now I'm going to turn it over to Jonathan, and thanks for joining us tonight.
All right, can you see my slide, Aviva?
Yes, I can see your slide.
All right. Well, I'm sorry my camera's not working, but we'll make it work. So I'd like to thank the Great Basin National Park Foundation and Aviva for inviting me to talk about Bonneville Cutthroat Trout tonight. As the park's fisheries biologist, like Aviva said, I've been lucky enough to work with Bonneville Cutthroat Trout here at Great Basin National Park for over 13 years. Throughout my presentation, I may call Bonneville Cutthroat Trout by several nicknames I have for them. The 3 most common would be just Bonneville, or I might shorten it up to BCT. And if I'm feeling really comfortable, I'll probably call them Bonnies. So if you hear me say Bonneville, BCT, or Bonnies, I'm talking about the Bonneville Cutthroat Trout. So the first point I'd like to make is that Bonneville Cutthroat Trout are not endemic to Great Basin National Park, meaning they do not just exist here. On the map, you can see the historic range in orange, which takes up much of western and northern Utah, some of southeast Idaho, a little bit of southwest Wyoming, and a little sliver of eastern Nevada. The second point I'd like to make is that Great Basin National Park is not the only agency that has been involved with Bonneville cutthroat trout conservation. 4 state agencies, Utah Department of Wildlife Resources, Idaho Fish and Game, Wyoming Game and Fish, and the Nevada Department of Wildlife are all actively involved with Bonneville cutthroat trout conservation, as well as several federal agencies, the Bureau of Land Management, United States Forest Service, United States Fish and Wildlife Service, and of course the NPS here at Great Basin. I'd also like to mention that the Confederated Tribes of the Goshu Indian Reservation have also actively restored BCT to streams they manage. And there are several non-government organizations that have played crucial roles in BCT conservation as well. Too many to list here tonight, but I'd like to mention the 2 I've been exposed to the most, which is Western Native Trout Initiative and Trout Unlimited. Both have done great work, not only with Bonneville, but with other native trout species throughout the West. And many of these organizations and agencies I've mentioned will often partner with universities. A good example of this is currently Great Basin National Park has an agreement with Brigham Young University to genetically analyze fin clips we'll be collecting this year. I'd also like to recognize and thanks the NDOW Ely office in Utah DWR, especially the biologist who works in the deep creeks, as these are the 2 agencies I work with the most, and we always appreciate their assistance. And lastly, sort of on my shout outs is Backcountry Horsemen of Nevada, specifically the High Desert chapter. They've been a crucial part of my current project and just awesome people to work with. Okay, so moving on to the presentation. Today, I will give you a little background on BCT habitat requirements, basic biology, and life history strategies. Then move on to the history of the subspecies, sort of told as a story, which will include a few different chapters. One on the formation of Lake Bonneville, the drying of Lake Bonneville, early interactions with humans, BCT conservation, current and future threats, tactics to combat these threats. And when I'm all done with that, hopefully I have time to go over some of the methods I used to sample Bonneville here and go over some of the projects I've worked on at the park. So we'll start off with a little background on Bonneville cutthroat trout. They are one of 14 subspecies of cutthroats. In this artist rendition of Snake Valley Bonneville, you can see the orange cuts or the slashes underneath its jaw. These are common in most cutthroat trout subspecies, and this is how they get their name. Bonneville cutthroat trout typically have large, round, black spots that are more concentrated near their tail. And they can have bright orange, pink, or red blotches on their side, on their gill cover, or on their belly. These colors are typically more apparent on mature fish during spawning or breeding season. In this photo here, you can definitely sort of see that typical spotting pattern that is more concentrated near the tail with fewer spots near the head. And on these next 2, you can see some of the orange to red blotches on the side and even on the gill cover. So Bonneville, like most trout, prefer cool, clean, well-oxygenated water. Water temperatures of 10 to 17 degrees Celsius are ideal for them. That's about 50 to 63 degrees Fahrenheit. Temperatures over 26 degrees Celsius or over 76 degrees Fahrenheit can be fatal to them. And they can definitely survive in temperatures much colder than 50 degrees Fahrenheit, but they'll develop and grow at a slower rate. They are very opportunistic feeders in small streams, and when they're young, their diet consists of a variety of aquatic and terrestrial invertebrates. Most common ones here at Great Basin National Park in our cold mountain streams are the caddisfly, stoneflies, mayfly, and several others. And they'll also eat worms and terrestrial insects such as ants and grasshoppers. In large rivers and lakes, they will also feed on small fish, and this allows them to grow to much larger sizes. These are pictures of Bonneville cisco, which are a large part of the diet of adult Bonneville cutthroat trout that reside in Bear Lake. All Bonneville need clean gravels and flowing water to successfully reproduce, and they need deep pools and other deep water habitat to survive the winter. And I'm finding this is especially true in our small mountain streams here at Great Basin. If the deep pool habitat is lacking, it is much harder for them to survive the winter. Moving on to their life cycle, eggs are laid in clean, silt-free gravel. If silt covers the eggs will suffocate and die. This is because oxygen from the water needs to diffuse into the egg, and carbon dioxide needs to diffuse out of the egg back into the water. In about 30 days, depending on temperature, Bonneville cutthroat trout will hatch from their eggs, and they'll have an external yolk sac still attached to their ventral side or their bellies. At this stage in life, they are less than 1 inch long and are called alevins. The alevins will stay in the gravel for about another 30 days, depending on water temperature. And once they emerge, they typically, or at least here at Great Basin National Park, they typically spend the first year of their life in slower moving, shallow waters, essentially trying to avoid the larger fish in the stream. Once they make it to this point in their life, their main job is just to survive and grow. And if they can grow until they reach maturity, they will spawn in the spring, typically on the downward side of the spring runoff. And once they spawn and the eggs are in the gravel, the cycle starts all over again. BCT are very adaptable and can exhibit a wide range of life history strategies in different habitats. Today, throughout much of the range, they occupy small headwater mountain streams. In these streams, they can fulfill their entire life cycle without migrating long distances. That is to say, they can find spawning habitat, rearing habitat, feeding and growing habitat, and overwintering habitat, all within a few hundred meters, or sometimes substantially less. I like to call this life history the stream resident. Other populations follow a fluvial life history. This is where the BCT will spend much of their adult life in larger rivers, and then will migrate into the headwater streams to find suitable spawning habitat. After hatching from their eggs, emerging from the gravel, and rearing in the headwater streams, the juvenile fish will head back to the river to grow to maturity. This is a more complex life history than the stream resident because it requires migrating long distances to reproduce. However, the positive trade-off is that the adults can grow to much larger sizes in a river than they can in the headwater streams, and this directly correlates to females being able to produce more eggs each time they spawn. The last life history I would like to go over is called adfluvial. Adults in adfluvial populations live, feed, and grow in a lake environment, and then swim upstream to find spawning habitat. Eventually, juvenile fish will return to the lake and stay there until they reach maturity. And again, this life history requires long migrations and allows the adult fish to reach much larger sizes. This life history today primarily exists in Bear Lake. So if any of you have ever been to Great Basin National Park or even nearby, it's easy to see that there is not much water here. And it's very similar in many areas of the Bonneville's home range. The Great Basin, after all, is a desert, which seems like an unlikely place for a salmonid to evolve, especially one that's known for evolving in a giant lake. To understand how this happened, we're going to have a little history lesson on the formation of ancient Lake Bonneville. I'm sure most of you know that one of the main features of the Great Basin is that none of the water in its streams or rivers flow to an ocean. They all flow inward into subbasins and either evaporate, sink into the ground, or flow into an existing lake. Well, from about 30,000 years ago to about 15,000 years ago, the Great Basin was a substantially cooler and wetter place. There was more precipitation, lower temperatures, and the lower temperatures also caused less evaporation. This all led to an increase in the amount of water flowing in streams and rivers throughout the Great Basin. And since there is no outlet to the ocean, all this extra water either caused water levels in existing lakes to rise, or it started forming new lakes in low areas. Lakes that form in this way, essentially the filling in of a basin due to an increase in precipitation or a decrease in temperature, are called pluvial lakes. And this was happening throughout the Great Basin. New pluvial lakes were forming, existing lakes were growing, and as nearby lakes grew, they often merged together to form even bigger lakes. And out of all the pluvial lakes in the Great Basin at this time, Lake Bonneville was the largest. And if anyone's curious, the time period, this time period is sort of a subset of the late Pleistocene. And Lake Bonneville can be seen in the upper right-hand corner of the map. So at its peak, somewhere around 15,000 years ago, Lake Bonneville covered much of western and northern Utah, extended into southern Idaho and eastern Nevada. Its total size was somewhere in the ballpark of about 32,000 square miles. To put this into perspective, it had a larger surface area than the largest of our Great Lakes, Lake Superior. It was also larger than 12 out of our 50 states. This is the time period I like to think of the golden days for the Bonneville Cutthroat trout. Not only did they have this massive lake to grow big in, but all the streams and rivers flowing into and creating this lake were great habitat for them as well. It would be reasonable to believe that at this time, all three life histories were simultaneously occurring and overlapping throughout most of the range, with some resident BCT never leaving the headwater streams, fluvial populations moving back and forth from rivers to streams, and adfluvial populations utilizing Lake Bonneville to grow to sizes that are far larger than any Bonneville is today. The adfluvial life history at this time was very important to the subspecies as a whole and promoted a good amount of gene flow throughout the entire population. I'd also like to point out that at its peak, Lake Bonneville extended into the Snake Valley, as seen on the maps in the section that I circled in red. So if you've ever been to the park, if you walked outside of Lehman Case Visitor Center and looked out over what is now the valley floor, you would have been able to see the waters and the shores of Lake Bonneville. And all the streams on the east side of the park would have flowed into this lake. So now we're moving on to another chapter, and this one focuses on Lake Bonneville drying up. So once Lake Bonneville reached its peak about 15,000 years ago, a natural dam breached and caused the Great Bonneville Flood. Water from Lake Bonneville was released into the Snake River watershed and flowed out towards the ocean. This dropped the lake level somewhere between 350 and 430 feet within a matter of a few days or a few weeks. For several years after the flood, water from Lake Bonneville may have still flowed into the Snake River watershed at a much slower and a more steady rate. However, soon after the flood, the climate began to warm and become drier. So from somewhere around 15,000 years ago to 13,000 years ago, right up until now, Lake Bonneville has been shrinking, and it actually became what is now known as the Great Salt Lake, and a few other remnant lakes such as Utah Lake and Sevier Lake. Sometimes on shorter presentations, I like to say that as Lake Bonneville receded, the Bonneville cutthroat trout sought refuge in the surrounding mountain streams. There's definitely truth to this, but sometimes I don't always like the picture it paints of fish swimming around in the lake, coming to the realization, hey, this lake's gonna dry up, and then making a mad dash up to the mountains. Obviously, this whole process occurred over several thousand years, and the subspecies slowly became less adfluvial and became more fluvial and stream resident dominant. Another impact of the desiccation of ancient Lake Bonneville was Bonneville cutthroat trout populations some of the Bonneville cutthroat trout populations became isolated from one another, which restricted gene flow and allowed certain regions within their native range to start diverging genetically from one another. The next chapter is on the interactions between humans and the Bonneville cutthroat trout, with a focus on post-European settlement. So even with the desiccation of what would today be the largest lake on the continent, BCT were still flourishing into the 1800s. They were an important food source for both Native Americans and early settlers that resided and passed through the region. The Goshute tribes who inhabited the Deep Creek Mountains north of Great Basin National Park called the Bonnies redfish and claimed they filled most of the small streams throughout the region. There are even stories of how Bonneville saved Mormon settlers and other early pioneers from starvation on more than one occasion. However, the interaction with early European settlers was not an advantageous one for the Bonneville, and soon after the area was settled by the first Europeans, the subspecies began to decline rapidly. I like to think of these being the dark days for Bonneville, and these dark days occurred for a little over 100 years, from the mid-1800s till the 1970s. During this time, Bonneville cutthroat trout were pushed to the brink of extinction. Water was being diverted from streams and rivers for agriculture and mining. Overgrazing increased erosion and decreased the amount of available cover, degrading their habitat further. Overfishing occurred at both a sustenance and commercial level, decimating some of the larger populations of BCT. One of the examples I found about a commercial fishery on Utah Lake claimed that in 1864, one net could produce as much as 3,500 pounds of fish, presumably much of that being Bonneville. And less than 70 years later, in 1930, only a single Bonneville cutthroat trout was caught during the entire fishing season. And then on top of all that, there's sort of the final straw, or what I call sometimes the coup d'etat for many of our populations of Bonnevilles: the introduction and invasion of non-native trout species. The three most common being brook trout, brown trout, and rainbow trout. Now, the reason that these three trout that were stocked heavily throughout the West are called non-native in the Bonneville Basin is because they were not originally from here, and they were actually brought to the region by humans. Brook trout actually are native to the East Coast, primarily the Northeast coast. Brown trout are actually native to Europe, and not even to North America at all. And rainbow trout are native to the Pacific coast, primarily the Pacific Northwest. So why were these trout so bad for Bonneville? Well, brook trout and brown trout compete with Bonneville, and they do so successfully that they've been known to displace them completely from a watershed. Rainbow trout can do about the same thing, but they're a little bit worse because they can hybridize with bonnies and dilute down their unique genetics. With all this going on, by the mid-1900s, it was believed that 100% genetically pure Bonneville cutthroat trout were probably extinct. So we're going to leave the dark days behind us. In the next chapter, I like to call it the era of conservation. And by the way, I'm just sort of making up these chapter names. If you Google era of conservation, it will not come up with anything, but just figured to make it a little more interesting. So this chapter sort of starts in the mid-1970s when one of the remaining populations of pure Bonneville cutthroat trout was rediscovered and an aggressive restoration program began. Individuals from that population were used to start new populations. Additional pure populations were also rediscovered. Non-native fishes were removed from several watersheds. Grazing and fishing became more regulated, and certain hatcheries even started raising pure Bonneville cutthroat trout for stocking. And all of this played a role in the Bonneville's historic comeback. And their comeback truly was historic. At some point in the 1970s, we only knew of one population of pure Bonneville, and today we have over 200. So now that the Bonneville have come back from the brink of extinction, what does their future look like? And I just like to mention that on this section, I'm mainly going to focus on Nevada populations because I'm more familiar with their current conditions and potential threats. And some of these threats that I mentioned may not be the biggest threats to other populations throughout different areas of their native range. So the climate is getting warmer, precipitation is more sporadic, and this coupled with decades of aggressive fire suppression is causing more catastrophic fires to occur throughout the Bonneville's home range. Here in Nevada, drought, wildfire, and increasing temperatures are probably some of the most serious threats to BCT today. This is especially true because our populations are small and isolated. We have had many success stories over the last 10 years in Nevada, but we've also seen these three threats wreak havoc on our populations. Hampton Creek, located in the North Snake Range, burned in 2014, and it is believed that the entire BCT population was extirpated. In 2016, the Strawberry Fire eliminated 85% of the individuals from the stream and reduced the Bonneville's distribution to just 1/4 of what it was in that stream previous to the fire. The current distribution of BCT in both Mill Creek and the lower perennial section of South Fork Big Wash has shrunk to less than half of what it was in 2019. And this is due to the current drought. And I believe the current drought is not only drying up lower reaches of streams that I've not seen go dry before in my 13 years here, but they're also limiting that deep pool, deep water habitat that's necessary for the Bonneville to survive the winters. Also, as the length of habitat gets shorter and the number of individuals in a given stream decreases, the risk of losing valuable genetic diversity increases. So what can we do to help protect the Bonneville in a changing climate? And again, a lot of these tactics that I'm going to talk about are better suited for small, isolated headwater streams, like we have here in the park and throughout Nevada. The first step I really think is just acknowledging that climate change is happening and take it seriously. Only once we do this can we decide which tactics are going to be best suited for the Bonneville into the future. One of these tactics I would like to call replicate populations potentially outside of their native range. To illustrate this, all the streams flowing to the east of the park are historically BCT streams, and all the streams flowing to the west side of the park are historically fishless. However, non-native fishes have been stocked in these west side streams. And if we could remove the non-natives from these streams, and even though they were historically fishless, put BCT into them, it would increase the number of populations and provide redundancy. These replicate populations could be used in the future to restock BCT streams affected by drought or wildland fire. I also want to point out that currently we do not have any projects going to actually do this. This is just a tool that we can think about and use as planning. If we did try to have replicate populations on the west side, there's 2 other streams that still support non-natives that we could turn into Bonneville populations. All right, moving on, assisted migration. So as the climate warms, ecosystems will tend to shift to higher elevations to remain in a temperature that best suits them. We've observed some species doing this at the park. Bristlecones and limber pines are moving up in elevations. And we've also noticed reptiles that used to only exist below the park boundary are now pushing up into the park as well. Fish will do this over time, and fish will definitely do this on their own over time. But if there's an impassable barrier, such as a large waterfall, then their migration up the mountain to cooler areas is over. That is, unless we give them a helping hand and move some of them above the waterfall. And that is the basis of assisted migration, moving at-risk species into areas that are better suited for them to survive, and moving species that would either migrate there naturally, maybe too slow, or couldn't get there because of a barrier. Next, I'd like to talk a little bit about genetic rescue. We have many small, very isolated populations that have had no gene flow to nearby populations for a very long time, and they're at risk of having decreased genetic diversity, essentially from an inbreeding depression. So in populations where genetic analysis confirms this is actually occurring, biologists can move individuals from one population to another to facilitate an artificial gene flow and increase their genetic diversity. The next tactic of better fire management, that one's really complicated, and I'm just gonna touch on it real briefly. But current and historic fire management has caused large buildups of fuels and an overabundance of climax communities. This is causing fires to be much larger and burn much more intensely than they did historically. And it makes it much more difficult for a healthy, early successional ecosystem to reestablish after a fire. And if these healthy ecosystems do not establish after a fire, it opens the door for many invasive plant species such as cheatgrass, which many of us know it does almost nothing for our native wildlife and fisheries. Lastly, I'd like to touch on public outreach, because this is a very important tool that we can use to further Bonneville cutthroat trout conservation. So thank you all for tuning in today. I really appreciate it. I'm also going to pause right here for a second and do a little mini outreach within this presentation. So I know I just talked about the potential benefits of replicate populations, assisted migration, and genetic rescue, which all involve moving fish from one location to another. But I urge you, please do not move fish from one body of water to another. In the state of Nevada, it is actually illegal to move a live fish from a water body without a permit. And there's just a lot of research and thought that needs to go into utilizing one of these tactics and moving fish from place to place to ensure that we do not do more harm than good, to make sure we are doing good. Just think about the rainbows, brooks, and browns. We move them around, and then we caused a big mess for the Bonneville and many other species. And we just got to make sure that we're not going to do the same thing again. But anyways, back to public outreach. The one area I'd really like to focus on that I hope public outreach and education can help is the public perception of native fishes, not just Bonneville, but essentially all the native fishes. I think there's some pretty good perception on natives. A lot of people like them. Hopefully you guys do if you're tuning in today. But there are still a lot of people out there that love their non-native fish. And for lack of better words, they hate certain native ones. You know, I spend most of my career working with them. So I look at a video of a Bonneville swimming around like this and I wonder how could people hate them? But I've been told multiple times that Bonneville are too weak and can't survive. I've been told their flesh is mushy and they don't taste good. And I've also been told they don't fight hard as a sport fish. I mean, we already know that they do survive well. They survived the desiccation of Lake Bonneville. They rebounded after 100 years of habitat loss and over exploitation. And they can still survive in lakes, rivers, and in tiny little streams no larger than a trickle. And personally, I do eat fish, and I have eaten Snake Valley Bonneville, and I have eaten Bear Lake Bonneville. And I think they're just as good as any other wild trout, and in my opinion, much better than recently stocked fish. So I think a lot of this dislike for natives and preference towards non-natives is rooted in personal experience, which to some extent I definitely can understand. So I grew up fishing in Western New York with my father for non-native steelhead. And even though I'm all about native fisheries nowadays, catching spring run steelhead in small New York State tributaries will always give me a feeling of nostalgia. And those memories will always have a special place in my heart. So I get it. A lot of people have a preference for what they grew up with, what they have a fond memory of, and for what they were taught by their loved ones. And for a lot of people, a recently restored native fishery is a completely new thing, and they have absolutely no connection with it. And even worse, we typically have to remove the non-native brook, brown, and rainbow trout to start our native fishery. So now the natives are directly responsible for destroying the fishery these people grew up with. Trying to win the hearts and minds of people that have these lifelong connections with non-native fish is a difficult thing to do. I think public outreach can help to some extent, but we also need to be understanding and respectful of their viewpoint. So the one area that I'm very hopeful that using public outreach and education can make a large change in public perception on native fishes, including the Bonneville cutthroat trout, is by making meaningful connections between the younger generations, especially of the younger generation of anglers, with them and their native fish. And I think if we can do this successfully, I think if we can make that connection, it'll help them sort of have that sort of love of natives throughout their life, and then hopefully they can pass that down on the future generations. If I grew up catching brook trout, which are actually native to New York, instead of steelhead, I know I still would have had just as many great memories fishing with my dad, and I probably would have a soft spot in my heart for those New York natives instead of steelhead. So hopefully moving forward, we can see a shift in the public sentiment that's sort of anti-natives. And honestly, I think it is already occurring, and we're already moving in that right direction. Most state game and fish agencies now have native fish slams, where they promote people to go out and catch all the native sport fish that the state has to offer. And Western Native Trout Initiative partnered with Rep Your Water and started putting native trout on apparel. And this was part of a campaign to promote not just catching your native fish, but also actually sort of being proud of the fish that are native in your region. This launched a social media trend. I believe it was #MatchTheHat, where people could post a picture of a Bonneville they caught while wearing their Bonneville cutthroat trout hat. And so with programs like this becoming more mainstream, I definitely think we're moving in the right direction, and the future looks bright. So to sum it all up, Bonneville cutthroat trout are true survivors. They evolved in a large lake, a lake larger than our largest Great Lake. They adapted and still flourished after that lake dried up. They came back from the brink of extinction after the species was decimated. And even though they're flourishing now, they're fighters, and they're still fighting for their survival. The future will definitely hold many challenges for the subspecies, but with proper management, the future still looks very hopeful and very bright. So now I'd like to take a little bit of time and talk about some techniques I use to sample Bonneville cutthroat trout here at the park and some of the projects I've worked on. So the main way we sample Bonneville cutthroat trout, and the main way people sample trout in most small headwater streams, is by electro fishing. We use a backpack electrofisher, which is essentially a battery with an interface where we can adjust the volts, frequency, etc. And then there's an anode. In this video that's playing, you can see the anode is the long yellow wand that I'm using to move through the water. And the cathode is a cable that drags behind me in the water. So essentially a giant battery that we can control the output of. Now, once I push down on the trigger on the anode, it will put an electric current in the water, and this can temporarily stun fish. Now, typically you have netters standing downstream of the person electrofishing with their nets in the water. And if the water is flowing fast enough, the fish will float down into their nets. If the fish is stuck in a slow moving pool or an eddy, then the netters will actively have to go after it. In the second video that I play, I'll play it twice. You may be able to pick out the fish being stunned and into flowing down into the net.
Coming down.
So it is located just upstream of the anode ring, and if you look for it right about here, you can see the fish pretty much loses its motion. It went down towards the net, and as soon as the electrofisher came out of the water, it regained consciousness, but still made its way into the net. So once we catch them, a lot of times we take length and weight data, and we have a little fish board, usually measure total length in millimeters, and then we will bring a scale out and measure their weight. And I just want to let you know too is we do our very best to make sure we don't harm any fish while we do this. They're usually just temporarily stunned, and typically they're not harmed or injured in any way. These fish were all just electro fish, and you can see them swimming around nice and happy in their bucket. And then once we're done taking our measurements, we get to let them go. So the next technique I like to talk about is eDNA, which stands for environmental DNA. This is a fairly new technology, although I think it's been around for almost 10 years now. It's a method of detecting a species' DNA in the environment to prove that they are either currently residing there or were there very recently. For fish, we essentially filter water and then send a filter off to a lab to be tested for fish DNA. This takes far less time than electro fishing and is a great tool to have, especially to monitor for non-natives. Another advantage is it's less invasive to the animal. We can detect if a fish is in the water without ever actually having to capture it and handle it. Now we're moving on to one of the probably the most important techniques, but also one of the most controversial, and that is Rotenone treatments. So Rotenone comes from a root from a plant in the legume family. There is a species of plant in South America that grows there, and the indigenous people actually use it. I believe there's a species of plant that contains Rotenone that grows in Africa, but I'm not sure. So the way the indigenous people have used it is a lot of times they'll actually grow this plant in their garden. And when they want to use it, they dig it up, smash the root up and sort of turn it into a pulp, put the pulp in the basket, take it to a river and put it in the water and let the Rotenone leach from the pulped root into the water. And Rotenone is toxic to fish. So when indigenous people do this, usually the fish become lethargic and much easier to catch. And the reason why they do this is they actually eat the fish. They've been doing this for thousands of years. The product I use is still actually extracted from a root. It is not 100% synthesized in a lab. However, we can make it more concentrated. And because the product we use is more concentrated, it just does not make the fish lethargic. It actually is effective at killing fish. One question I get asked a lot, can the people eat the fish after they're dead? You cannot. When you treat a stream with Rotenone, humans cannot eat the fish. It is a pesticide that's heavily regulated by the EPA for good reason, just to make sure we keep everyone safe. On another note too, Rotenone can actually be deactivated with potassium permanganate. So if you want to stop treating a stream at a certain point, you can set up a deactivation station. So throughout the presentation, you probably did hear me say non-natives were removed, or you need to remove non-natives. This is typically how we do it. It's the most effective and most efficient way. And another good point to make, if a deer walks down to the stream during the treatment and drinks the water, the deer will not die. However, it is very, very toxic to fish and other animals that have gills, such as amphibians. Luckily, here at the park, we did not have any amphibians to worry about. So on a more positive note, typically after Rotenone treatment and all the non-natives being removed, we get to do reintroductions. And so here in Nevada and in the park, we do not have a hatchery that raises Bonneville. So to start a new population and do a reintroduction, we actually have to use fish from existing populations. So we go out with electro fishers, capture fish, load them up on trucks with big tanks equipped with aerators and oxygen gas, drive that truck to the trailhead usually, and then we pack them out, many times on our backs. Recently, luckily, we've actually been able to pack them on horseback and on mules. I'd also like to mention almost every Rotenone treatment we've done, maybe everyone, and the reintroductions I've done, I've always worked with Nevada Department of Wildlife. So they play a crucial role in our management as well. So those containers on the side of that horse or that mule, the ones that are white and yellow, those are full of fish. And that animal actually took it up to Johnson Lake for us when we introduced fish there. So I've been very fortunate to meet a group of people, the backcountry horsemen, that have really helped us out with that. Genetic analysis. This is another useful tool to make sure our Bonnevilles are still 100% pure. We can take a tiny little clip off the caudal fin, which is their tail fin, and then we can have a lab genetically analyze it. And it's just really good to know that before we start another population, the one that we're going to use as the donor source, the source, is actually 100% genetically pure Bonneville. All right, so from this point forward, I just figured I'd talk to you a little bit about the projects I've worked on. The very first project I worked on was one that we essentially were going to resample every single BCT stream in the state of Nevada. We collaborated with the Nevada Department of Wildlife, and it was just a really awesome summer. I got to see almost all the park and a lot of places outside the park, and it really showed me how amazing Nevada could be, and it's probably one of the reasons I'm still here. But three main important things came from that study, or that project, and it was, number one, NDOW actually found a new population of BCT. Well, at least they documented a population that we previously did not know about. The second thing was all our populations came back 100% pure, which was great news. And the third one wasn't as great a news, but it was still really important to know that we had brook trout in the Bonneville Cutthroat Trout Restoration Area on Snake Creek within the park. So after we learned this, our second project was to remove all the non-natives from Snake Creek and release Bonneville Cutthroat Trout back into Snake Creek. We were fortunate enough to get funding to put a fish barrier near the park boundary, so we could do this in both upper and lower Snake Creek. And we also were able to salvage as many Bonneville as we could from the upper section before we treated it with Rotenone. And we salvaged 450 non-native brown trout, and NDOW used those in a recreational fishery that is just in a man-made reservoir where there's no natives. And so we got to actually save as many fish as we could before we treated the stream with Rotenone. We released Bonneville in 2019 in Snake Creek, and I'm excited to try to do a population survey this fall and see how they're doing. And then that sort of brings us on to our current project, where we are trying to establish Bonneville Cutthroat Trout in Johnson and Baker Lakes. Both lakes are high elevation, over 10,500 feet. That area of the park gets the most precipitation, and has the coolest temperatures. So we were really hoping that it would be refugia as the climate warms and becomes drier. That's the project of the Backcountry Horsemen are helping us out on, and NDOW is still helping us with that one as well. We already released some Bonneville and Johnson Lake, and we're currently trying to remove non-natives from Baker Lake. At that point, I think I am ready for questions. If you guys want, you can watch one more little video of Bonneville. I always love underwater videos, but would you like me to come in?
Yeah, John. Why don't you just come on in here and we can be together on this one screen, and we'll go with some questions. So if anyone in the audience has, there's a few questions here, but if you have any more, throw them in the Q&A, and we will get them answered.
Okay. Do you want me to stop sharing my screen real quick?
Yeah. But then you can just, once you stop sharing, just come on over, and I think that will be fine. Great job. Thank you so much. I think we all learned a lot. And see, superb presentation. So thank you. So I'm going to start. Well, there is a question about, and this, you're the fishery biologist, so this may not be within your wheelhouse, but a question was about if there's any move to reintroduce beaver to the area so that they can provide deeper and cooler water. Do you know anything on that level?
So currently right now in the park, we do not have any plans to reintroduce beaver. Yeah, I'm going to leave it at that. There are beaver off the park, in the surrounding area, and there's a lot of good research coming out about how beavers can help different watersheds and the fish that live in them. But currently, right now, we do not have any plans to reintroduce beaver.
So maybe in the future, who knows? A question about the species. Is the species elastic in its ability to be fluvial, adfluvial, etc.? In other words, if moved to a different environment in an attempt to expand their range, will they change their preference? Yeah.
Yes, I don't have any direct evidence of this, but typically that's just how trout work. I can give an example with Lahotans. I believe there's been examples where Lahotans from headwater streams, remnant populations where people didn't even know they still existed, were taken back to large lakes, and then they became that adfluvial sort of lake population. So they definitely have the ability to do that. And here in the Great Basin, we pretty much only have the stream resident because our streams are so small and isolated. So I don't do a lot of work with that, but the way I've seen other cutthroat trout be able to, if you move them to a different habitat, the way they can adjust quickly is pretty amazing.
Yeah, makes sense. Question about if there's a way to remove non-natives from a lake. I assume it's probably the same as a stream, but I'm not sure.
Yeah, so there's again, the most effective and the most efficient way, both time-wise and money, is to use Rotenone or a different pesticide. But at this point, Rotenone is pretty much the only one that's in general use. And it will essentially, yeah, it works the same way as in the stream. You can put it in the lake and it'll kill the fish, and then Rotenone does break down naturally in the environment. UV light breaks it down, so the sun will. Oxidization breaks it down, and so just over time, it'll just naturally get removed from the lake.
Right, but I would imagine anywhere that, like you said, has amphibians is going to be an issue. So
yes,
we'll probably use some different tactics.
So if I was in charge of trying to remove all the non-natives from a lake without using Rotenone, I would definitely go heavy on gill nets, especially for trout. There are other like traps and stuff you can use, but I feel like gill nets are probably the most effective for trout, if that's what your non-native is.
Mm-hmm. Hey, let's see if we have any more. A couple of great jobs there. I'll ask a question, and if anyone else has questions, throw them in the Q&A. But I was curious when you talked about how the lake breached the dam and float. Do we have any idea what made that happen?
Yeah, and this isn't my wheelhouse, but on the research I've done, I've sort of read one story like where it finally just got high enough and it started trickling over the dam, and then it quickly eroded the dam out. Essentially, it was two alluvial fans. So it wasn't like a solid bedrock or anything dam like that. It was just a deposition on a geologic time scale. I read another thing that said because it was alluvial fans, there was probably actually seepage going underground and softening that all up. So once it did start to cut down, it was already, it wasn't like solid, good soil. It was already sort of wet and muddy underneath. So it just blew out really quickly.
Very interesting. All right. Well, I think we have another question here. So a question about Strawberry Creek. The restoration project on Strawberry Creek should be well on its way to completion. Are you seeing increased BCT? Probably the problem was the fire, right?
Yeah, so the fire restoration on Strawberry Creek, we are putting in post-assisted log structures and beaver dam analogs. Essentially just they just look like wood jams, just like a bunch of dead wood just caught together in a stream. And the purpose is different in a few different locations. Some places we're actually trying to catch sedimentation as erosion comes off the hillsides. Other areas, the channel is really narrow and incised, and we're actually trying to cause erosion there and make the channel wider. So when high flows come through, it's not as powerful. If you put the same amount of water on a little area, it'll be much more powerful. If you can spread it out, it'll be better. And in a few spots, we also, all that'll help the riparian habitat, which will in turn help the Bonneville cutthroat trout. And the one structure that we built that was the actual beaver dam analog that immediately formed a pool, that one sort of had the most essentially immediate return on your investment. And within the next spring, we did that last fall, and within this spring, there was already Bonneville using that. It's still a little too early, so we just installed these in-stream structures in the fall, so it hasn't even been 1 year. So it is a little early to actually say like, hey, we're getting a big increase in population numbers, but we have seen Bonneville use the structures we've installed, and I think everything's looking real good there, and I'm real hopeful in the future. Since we installed the structures, I haven't done a population survey either. So we're gonna do one in late September, and we'll have a better idea then.
That's interesting, all that stuff you did. I had no idea.
That one is not a fishery specific project. I'm actually not even the lead. I'm just been
Okay.
helping out. So there are a lot of other aspects to that, which
Right.
include seeding, weed treatment, a bunch of other things sort of up on the landscape. And our biologist, Margaret Horner's been in charge of that. And I just try to help out as much as I can with the stream side of things, the in-stream structures.
Really interesting. And a question on the average lifespan of the Bonneville.
So this definitely varies from place to place, just like their life histories. It's pretty common to think that, 7 years of age is pretty standard for a small, cold headwater stream. However, in Bear Lake, which is it's right on the border of northern Utah and Idaho, that is the only main adfluvial population I can think of off the top of my head, where they grow big in that lake and then, migrate up to spawn up the rivers and streams. They have a much longer lifespan. Some of them do not even reproduce till after the age of 5, and it's just a different habitat for them, and they grow a different way, and it increases their lifespan much longer.
So the ones, let's say in the park, those would last for like 7 years?
7 years is a
And
really
then
good average.
They also asked, the attendee also asked how many times they might spawn during that time. Like is it the first year they're starting to spawn already?
So males generally mature a little bit earlier than females, just because essentially eggs are more expensive to produce. It takes up a lot more energy to make a bunch of eggs. So our best guess is the males may be able to reach maturity 2 to 3 years, and the females are probably reaching maturity in 3 to 4 years.
Oh, I wouldn't have thought it was so long. That's interesting. And we have a bunch of people saying thank you for your program and for your work. Historically, were there other native fish that existed locally?
There was many different fish that lived in Lake Bonneville. Definitely do not quote me on this, but it might have been as many as like 28 species. I don't know off the top of my head, but in Bear Lake, there's Bonneville Cisco, Bonneville whitefish. Here locally, right around the park, we probably had some native sculpin. There was 3. There's a good chance that sculpin, speckled dace, and maybe a shiner were also native. And before I got here, the park did in 2008 and earlier release some of these non-game natives into the park. However, they did not seem to take that well, so we haven't retried. They're sort of more evolved and more suited for warmer water and not as violently flowing water, a little bit stiller, and just yeah. Our mountain streams are pretty cold and pretty fast flowing.
Yeah, well, I can imagine up at 10,000 feet. I mean, that's pretty impressive. So 2 more questions. One is just about if there's any other way to tell the species besides the spots. I mean, you said the little red.
Yeah, so to tell visually, to tell Bonneville from other cutthroat species is pretty tough. But from rainbows, I like the spotting pattern. They're usually larger, rounder spots and less by the head. If you did notice in that picture I had of the rainbow, there were spots all from essentially evenly distributed and a lot more of them. And the edges on the rainbow are typically more sort of irregular and jagged, almost like pixelated. The slash, like Aviva mentioned, that's generally a sign of a cutthroat. If it's a half rainbow, half cutthroat hybrid, they'll also have those cuts typically. And yeah, that's pretty much the main way to tell them apart from rainbows. There's a lot of big differences from brooks and browns. Brown trout have sometimes, usually in their stream form, have red spots with blue halos. Brook trout also have red spots with blue halos, and brook trout have lighter spots on a dark background.
Well, one last question, which is oh, actually there's two questions now. Is there a place where people can follow the status of these projects? Because they're really interesting projects. So I think people would like to do that.
So from time to time, we, myself or one of my colleagues will write an article in the Midden, and that is on our website. It's the Midden is essentially the natural resource newsletter. So everyone in natural resources, and I'm sure cultural is included as well. If we want to write an article, we can, and it's open to the public, and the public can actually download it off the internet and then read it. Another place would be a little, yeah, the Midden would be the best bet. But these are SNPLMA funded projects, and there is ways to look up different SNPLMA reporting as well. But that probably will not have the details you're looking for.
Well, we'll keep you updated. The foundation, one of the things we've done just recently, because the Midden is totally amazing publication, is that we've taken like each article and we're going to spread them out like one a month, and then have them like Facebook posts that take you there. So try to get this information more out to the public. But we're just at an hour, and thank you so much. This was really wonderful. I know that the audience appreciated it. And thanks everyone for joining us. This has been a really fun lecture series. I appreciate everyone who's been attending, and we just are so happy to have you, and have a very good night. Bye.